To all attendants, let me introduce myself. My name is Novia Rachmawati. With our teams, we will present you our research about Colchicine Mediated Polyploidization, a potential way to enhance carbon capture capacity of Euglena gracilis. Recently, greenhouse gases, particularly carbon dioxide, have significantly contributed to global warming. This has led to rising temperature and notable climate change impact across the globe. As global awareness of this issue, we need to find practical solutions to reduce carbon emission and their adverse effect. Among the various carbon capture techniques, microalgae-based systems are gaining significant attention due to the high efficiency. Microalgae can sequester carbon dioxide at a rate 10 to 15 times faster than the higher plants. Specifically, Euglena, a type of microalgae, can capture between 0.03 to 0.06% atmospheric carbon dioxide. This makes it an effective candidate for carbon capture initiative. To further improve Euglena carbon capture capability, several strategies are being explored. One such strategy involves using colchicine for polyploidization, which increases the number of chromosomes and potentially enhance carbon uptake. We collected the sample from Dieng peatland Wonosobo Regency, Central Java, Indonesia, in triplicate to ensure accuracy. After collection, we isolate and purify the Euglena gracilis cell to separate them from other microorganisms. We then transfer a single cell culture into microplate and expose them to colchicine solution. We test 5 different concentrations of colchicine over 3-time interval from 24, 48, and 72 hours. After the treatment period, we subcultured the cell and conducted a thorough evaluation. We assess various factors including survival rate, cell area, DNA concentration, growth pattern, biomass, and growth kinetics. Now, let's define into the preliminary result of our study. We focus on 3 key factors such as survival rate, DNA concentration, and cell area. The survival rate is a crucial indicator of how the cell can endure under specific experimental conditions. Our findings reveal that both increasing the colchicine concentration and extending the incubation time have a notable impact on the survivability of Euglena gracilis. Additionally, polyploidization induced by colchicine treatment has shown promising results. It can lead to an increase in both genome size and cell size. Based on our observation of survival rate, DNA concentration, and cell area, we identify 6 optimal candidates, such as 0.1% and 1% concentration at 24-hour induction, 0.5% and 5% at 48-hour induction, and 0.5% and 5% at 72 hours induction. So, in the first result about biomass, our result showed that Euglena gracilis treated with 0.1% and 1% treatment achieved the highest dry weight and the greatest biomass of productivity. This outcome correlated strongly with the increase in cell area observed in this treatment. The larger cell contribute to accelerate dry biomass and enhance biomass productivity. It's also important to note that the cell biomass is influenced not only by cell area but also by photosynthesis and cell cycle. These factors together play a crucial role in determining overall biomass in Euglena gracilis. Moving to the specific growth rate, cell density, and doubling time, on the growth parameter, we observe this treatment has positive result with the peak occurring on day 6. However, our analysis of the specific growth rate revealed a decrease in the rate of cell population per day for the 0.1% and 1% colchicine treatment. This resulted in a longer doubling time, indicating that this treatment required more time to mitosis to complete. This observation aligned with findings from studies by Minso et al. And Nizat and Mansouri. Their research suggests that ploidy can inhibit microtubule formation during mitosis, leading to extended mitotic times. And the last one, about growth kinetic, It's to understand the life stage of Euglena gracilis. Additionally, this model helps us determine the optimal culture system for enhancing the capability of Euglena gracilis as a carbon capture agent. Our results demonstrate that the compared and logistic growth model are particularly effective in describing the growth kinetic of Euglena gracilis. This is supported by R-square value reaching up to 0.9, indicating at high level of fit for these models. So, based from our analysis, we can conclude that treat Euglena gracilis reveal higher dry weight, biomass productivity, cell density, specific growth rate, and doubling time compared to the wild type. The enhancement of this parameter suggests a potential correlation between polyploid Euglena and its effectiveness as a carbon capture agent. So, for our next project, we will take a comprehensive studies on the factor that regulated carbon capture capacity, such as transcriptome analysis, carbon capture efficiency, and cultivation system. Based on our current findings, we are highly confident that this research will yield valuable insight and prove to be a significant asset in advancing carbon capture technologies. Thank you for your attention. We look forward for sharing more about our progress in the future. Euglena polyploids enhancing our potential to enter our future life.